Hello students, so today we are going to learn the second part of the chapter Inheritance and Variation. In this part of the video, we are going to learn Monohybrid Cross, Dihybrid Cross, Mendel's Law of Inheritance. There are three laws: Law of Dominance, Law of Segregation, Law of Independent Assortment, Back Cross and Test Cross, Neo-Mendelism, in which we are going to learn incomplete dominance, complete dominance, multiple alleles and Pleiotropy. Further, we are going to see few animation images. Here we can see incomplete dominance, that is pink colored flower and white flower. When is crossed over, we get purple colored flower. Then sickle cell anemia, where the RBCs we can see sickle shape or C shape, and here the scientist Mendel. Now we learn the first part, that is monohybrid cross, a cross between parents differing in only one heritable trait is called monohybrid cross, as the term mono means single, one. So here in monohybrid cross we are learning about one trait. Example, cross of pure tall and pure dwarf plants. Second point, Mendel performed the monohybrid cross between two pea plants with only one pair of contrasting character. So here we can see the monohybrid cross between pure tall and dwarf plants where only one character is considered. You can see in the animation a cartoon character which helps to understand this concept very clearly, where capital Y small y, capital Y small y. When crossing over takes place, capital Y, the pure tall, whereas capital Y, capital Y, small y, hybrid one, and here small y, which is pure dwarf. So you can see this in case of yellow color or tall, where you can get the ratio 3:1, that is phenotypic, or 1:2:1, that is one pure tall, pure dwarf and two hybrid ones. Then the image that is for monohybrid cross parental generation, we can see TT for tallness, small t for dwarf, which are the gametes capital T and small t. When crossing over takes place, we get in the F1 generation genotype all are capital T small t, whereas phenotype all are tall. So here we can see capital T small t, capital T small t. The gametes will be capital T and small t, capital T and small t. In the F2 generation, when these are crossover, we get capital T, one pure tall, capital T small t, the hybrid one, capital T small t, again the hybrid one. But these both will be tall, reason dominance, that is the capital T, since it is dominant, the feature seen will be dominance, whereas the fourth one, that is small t will be for dwarfness. So here one pure tall, one pure dwarf and two hybrid ones. In case of phenotypic ratio, it will be 1 2 3 3:1, whereas in phenotypic that is in case of genotypic, it will be 1:2:1. So as you can see in this case, that is the animation how the monohybrid cross can take place. Now we come across the dihybrid cross, the second type. A cross between parents differing in two heritable traits is called dihybrid cross. So here we are considering two traits. Example, cross of pure tall, rounded seeded plant with dwarf, wrinkled seeded plant. Mendel also performed the dihybrid cross between pea plants that differed in two pairs of contrasting characters. As we can see, tall, rounded and dwarf, wrinkled. So here we can see various combination with a simple animated image showing capital R, capital Y, capital Y, capital R, capital Y, small y, capital R, small r, capital Y, capital R, small y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y, small r, small y, capital R, small y. These are the combination which we can see. Now here in this image of dihybrid cross, we can see the parental one P, capital Y, capital R, small y, small r. In the F1 generation, we get the phenotypic ratio as capital Y, capital R, small r, because the dominant one being the rounded seed plant. Then we can see here in the F2 generation, the yellow rounded, the dominant one. Here we can see the gametes capital Y, capital R, small y, capital R, capital Y, small r, small I in horizontally as well as vertically the either parental gametes. Then when the crossing over takes place in the F2 generation, by this checkers method of Punnett square, we can see the formation of dominant ones, which is we can see the inverted L and across, that is diagonally, and 1, 2, 3, which is the second one where it is in the ratio of 3. Then we can see here again the yellow wrinkled one, the lower again smaller L shape, and lastly one single pure dwarf small y, small r. So we can get the ratio, that is the diagonally and inverted L, that is 9, and here the green rounded one which is 3, and then yellow wrinkled one again 3, and lastly single one dwarf. So we get the ratio 9:3:3:1. That is in case of dihybrid cross. If you count all in all, that is 9 plus 3 12, 12 plus 3 15, 15 plus 1 16. In all, we get the number of offspring which is present in the checkers method. Now, moving further, we come across the Mendel's law of inheritance. Mendel proposed 3 basic postulates on the basis of which 3 laws were formulated. These are described below. First, the law of dominance. In monohybrid and dihybrid crosses, the phenotypic character are controlled by discrete units called factors, which was coined by Mendel, because initially it was not known that these are nothing but genes. Further, in a dissimilar pair of factor, one member of the pair dominates, that is dominant over the other recessive. So as we saw in the phenotypic ratio, the dominant one which is seen in the offspring, the characteristics, whereas the other character dwarfness is present but not seen or not expressed, which is termed as recessive. The law of dominance is used to explain the expression of only one of the parental character of a monohybrid cross in F1 and the expression of both in F2. So we can see one character which is dominant, which is expressed in the F1 generation. In the F2, both are expressed but in the ratio 3:1. Then, statement of law of dominance, which states when two homozygous individuals with one or more sets of contrasting character are crossed, the alleles, that is the characters that appear in the F1 generation are dominant, and those which do not appear in the F1 generation, they are recessive. As I mentioned, the recessive characters are present but it is not expressed, it is suppressed. Clear? Now you can see the image. Tall, capital T, short, small t, in the F1 generation, after crossing over, we get capital T, small t, small t. In the F2 generation, we get capital T, small t, and small t. So, here we can see 3 phenotypic ratio, tall as the dominant character and one dwarf. But when we consider the genotypic ratio, it is capital T is 1, two hybrid ones, capital T small t, and one pure dwarf, that is small t, which is Mendel's law of dominance. Then another image which shows law of dominance, three genotypes, capital Y, capital Y, capital Y, capital Y, small y, and small y, small y. So in three we can see yellow color or the tallness which is dominant, and one which is dwarf or recessive gene. So three yellow and one green. So one trait is dominant over the other. Now here, if I take an example, in a family, if there are four child, brothers, sisters, out of which we can see three might be either tall or short, one may be either short or tall, vice versa, or there may be difference in the color or difference in the hair shape, smooth, curly. So that pattern may be seen even in case of humans. Then coming to the second law of segregation, otherwise called as law of purity of gametes. This law is based on the fact that alleles do not show any blending or mixing, and both the alleles, that is characters, are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. As we learned in the first law, one which is expressive, which is seen, whereas other which is suppressed or recessive trait is not seen. So dominant one is seen and the recessive not seen, and it is clearly observed in the F1 generation. They don't mix or blend with each other, or in other words, as the law suggests, segregate or separate clearly. Second point, during formation of gametes, these two alleles or factors obviously separate or segregate, otherwise recessive type will not appear in F2 generation. The gametes which are formed are always pure for a particular character or trait. A gamete may carry either dominant or recessive factor but not both. That's why it is called as law of purity of gametes. So as mentioned over here, the character which is seen or observed are the dominant ones, and the other one is the recessive one, and both are always pure. As mentioned in the first point, they do not blend or mix with each other. The statement of law of segregation states that when hybrid F1 forms gametes, the alleles segregate from each other and enter in different gametes. So the alleles or the factors, they segregate, or in other words or simple words, separate from each other. Further, the gametes formed are pure in that they carry only one allele each, either dominant or the recessive allele. Hence the law is also described as law of purity of gametes. So since the dominant which is observed are purely tall or purely dwarf, it is also termed as law of purity of gametes or the law of segregation. Clear? Now observe the chart over here. The parental generation were pure red, pure white. So here in the first generation, as per the law of dominance, all four will be pure red. Okay, so that is hybrid red, but since R is dominant, so it will be red. In the second generation, when crossing over takes place, we get three red and one pure white. Again, that is it will be in the ratio 1:2:1, but since the hybrid ones, they are dominant, they will be observed, and one recessive trait which will be white. Now moving further to the third law of independent assortment. This law is based on dihybrid cross. That is, in the initial first two, as we observed the two laws, law of segregation, law of dominance, both with relation to monohybrid cross, but the third law is purely based on the dihybrid cross. So here it is based or basic principle of genetics developed by Mendel. It describes how different genes or alleles present on separate chromosome independently separate from each other during formation of gametes. These alleles are then randomly united in fertilization. In dihybrid cross, F2 phenotypic ratio is 9:3:3:1 as we saw in the dihybrid ratio by the checkers method. Then indicates that the two pairs of characters behave independent of each other. It can be concluded that the two characters under consideration are assorted independently, giving rise to different combinations. As we saw in the dihybrid cross, again we can see here the ratio 9:3:3:1. That is, in the F1 generation, as we get capital A small a, capital B small b, crossing with again the hybrid one, capital A small a, capital B small b, we get 8 different combinations. And when this is crossed over each other in the F2 generation, we get, as I mentioned, inverted L and the diagonal part, which is in the ratio 9, that is yellow round. And then we can see over here alternatively 1, 2, 3, which is yellow but wrinkled. Then 3 over here, that is green round, and 1 pure green wrinkle. So 9:3:3:1. So statement of law of independent assortment states that when hybrid possessing two or more pairs of contrasting factors or alleles forms gametes, the factor in each pair segregate independently of the other pair. Now, again, few animation to understand monohybrid, dihybrid and all the three laws in a very simple pattern, or how we can solve the puzzle of checkers board or Punnett square. So you can see the first animation here, capital A small a. So when the crossing over is done, capital A, then capital A small a, capital A small a, small a, small a. So we get 1:2:1 in genotypic ratio, or 3:1 in phenotypic ratio. Similarly, second one, here it is capital B small b, capital B small b. Again, when you cross over, we get capital B, that is capital B small b, capital B small b, and small b small b. So again, it is one pure, one hybrid, one hybrid over here and second hybrid over here, and one pure dog. So it is 1:2:1 in the genotypic and 3:1 in the phenotypic. So this is about the monohybrid. Now when you see the dihybrid cross by checkers method or Punnett square, it is capital A small a, capital B small b, crossing over with hybrid one, capital A small a, capital B small p, and the gametes capital A, capital P in the either side, capital A small p, small a capital P, small a small p. When the crossing over is carried out, we can get again in simple way, that is inverted L, and diagonally we get the ratio 9, and here alternately 1, 2 and 3, that is the second dominant one, and the thirdly over here, that is small a, capital B, and the other two small a, capital B, small p, same over here, small a, capital P, small p, and lastly the single ratio small a, capital small p, the pure dwarf one. Clear? So this animation explains us both monohybrid, dihybrid and all the three laws. Now, moving further to back cross and test cross. Back cross. The F1 individuals obtained in a cross are usually self to get the F2 progeny. So, from the parental one, we get the F1 individuals. Then F1 individual, when it is crossed over, we get the F2 individuals or the F2 progeny. Further, they can also be crossed with one of the two parents from which they were derived, either recessive or dominant. Such a cross is known as back cross. So, when you get the F1 individuals, now this can be again derived, that is by using either recessive or dominant, we can again cross over. So, we get nothing but back cross, or which is called as back cross. So you can see a chart which will help us to understand much better. F1 hybrid, capital T small t, now which is usually what we saw in the monohybrid, dihybrid, or in the three laws. When the hybrid ones, when we get in the F1 generation, or in the parental, it is capital T, small t, dominant recessive. When it is crossover, we get the hybrid ones, capital T small t. But here, what is done is capital T small t is crossed over dominant or with the recessive one. Dominant, capital T, recessive small t. So you can see here, the gametes formed is capital T small t in case of hybrid, but in case of dominant, both are capital T. Similarly, in recessive, both are small t. So here, when you cross over in the F2 generation, we get all four as tall, that is two pure tall, two hybrid ones, but again it is dominant. Whereas in case of crossing with the recessive one, we get 50% tall, that is hybrid ones, and 50% dwarf, that is the recessive trait. So here it is 50-50. Here it is full 100%. But when this is F1 generation, it is crossed over the parental one, either dominant recessive, it is called as back cross. Clear? So that is why F1 is crossed over dominant, F1 crossed over with recessive. Now moving further, test cross. The cross of F1 hybrid with the homozygous recessive parent is known as cross or test cross. Now here it is only with the recessive parent, small t, not both. So if it is only with the recessive parent, then it is called as what? Test cross. It is used to test whether an individual is homozygous pure or heterozygous hybrid. So such experiment helps to identify it is a homozygous or heterozygous. Test cross is easy, simple, repeatable and predictable. So we can predict what is the outcome. It can be repeated as many times as you want. It is very simple to carry out and easy method. Next point, test cross can be used to find out genotype of any plant with dominant expression. So you can be easily identified which expression is dominant. But it is not known whether it is homozygous or heterozygous, that is pure or the hybrid one for that trait. For example, a pea plant having violet, that is purple flowers, is crossed with a pea plant with white flowers. If all the flowers produce a violet, we can conclude that the plant is pure or homozygous, and if we get violet and white flowers in the ratio 1:1, we can conclude that plant is heterozygous. As we saw in the previous one, that is in case of back cross, how the crossing over gives you either pure or the 50-50 hybrid ones. So test cross is also used to introduce useful recessive traits in hybrids or self-pollinated plants during rapid crop improvement programs. Following is a graphic representation of test cross. Recessive parent is crossed to find out unknown genotype. Now, another graphical representation of test cross to make the understanding much more clearer. So here you can see homozygous recessive, when crossed over, we get the gametes as smaller, here also smaller, as both are recessive. Then, if it is crossed over with dominant phenotype RR, and here capital R smaller, the hybrid one, all flowers are violet, as we read earlier. Unknown flower is homozygous dominant. In this case, we can see it is 50% violet color or purple color and 50% is white. So half of the flowers are violet and half of the flowers are white. Unknown flower is heterozygous. Okay, so it is a simple graphical representation showing the test cross. Now, further, deviations from Mendel's finding. What is deviation? Changes or small variation which is taking place. So what Mendel told, all the laws from which there are small deviation. Now what are these? Few generalizations were arrived at by Mendel on the basis of his experiments of garden pea plant such as single trait, single gene, two alleles. Second, two alleles show interaction in which one is completely dominant, as we had seen, dominant or recessive. Factors, that is genes for different traits present on different chromosomes assort independently or segregate, as we learned law of independent assortment. With the passage of time, the number of deviations were observed, identified in the post-Mendelian era that gave additional information on the patterns of inheritance. That is, later on, as more experiments carried out after Mendel had carried out experimentation after his experiments which was seen or observed by the different scientists. These deviations then described as Neo-Mendelism, that is different or new from what Mendel has mentioned. Just a image I put up to show four different colors of pea plant. Now, further, it was observed that phenotype expression of a gene can be modified or influenced by the other gene. These genes interactions of two types. So it can be said that the phenotypic expression of the gene can be changed, modified or influenced by the other gene. So it is put into two categories. One is intragenic interaction or intragenic. Intragenic, as the name says, within. Intergenic, by the other genes. So here occurs between the alleles of the same gene. Example, incomplete dominance and codominance. It also occurs between the multiple alleles series of gene. Then intergenic interaction occur between the alleles of different genes present on the same or different chromosome. Example, pleiotropy, polygenes, epistasis, supplementary and complementary genes. So these are different observation which was seen, so which we are going to learn further. Some of the interactions are discussed. So here as a small chart is given, do you know the deviation? As we just read out earlier, single trait, single gene, two alleles showing interaction like codominance and incomplete dominance. So what is co and incomplete? We learn in the next slide. Single trait, single gene, more than two alleles, then multiple allelism. Single trait, more than one genes, which is polygenic inheritance. Poly means many, showing different epistatic interactions or additive effect. Then single gene influencing many traits, which is pleiotropy. Again, what is that? We learn further. Now, incomplete dominance. So first one we take incomplete dominance. Now in the incomplete dominance, both the alleles, that is genes of an allelomorph pair, express them partially, means half. So whatever we learned in Mendel, that is dominant recessive, here it is incomplete, means the character which is not expressed completely but partially. One allele cannot suppress the expression of other gene completely. So in Neo-Mendelism or in this incomplete dominance, one gene is not dominant over the other. So it cannot suppress the other expression of the gene. In such case, there is an intermediate expression in the F1 hybrid. A well-known example is the flower color of Mirabilis jalapa, as I shown over here, and even in the second slide as shown you the animation where the pink colored flower crossing with white gives you purple color, right? Or red and white giving purple. So here if a red colored RR plant is crossed with a white color smaller, the F1 offspring have pink colored flowers. Result, genotypic ratio 1 RR, 2 RR, R, that is capital and small r, and 1 small r. In phenotypic ratio, it will be 1 red, 2 pink and 1 white. So if you can see in the chart, that is parent red color, white color, capital R, small r, gametes, capital R and small r in case of white flower, F1 hybrids, capital R, small r. Now, F2 generation, that is selfing of F1, we get RR red, R pink, then RR, there's capital R, small r, pink, and smaller white. So we get here it is 1:2:1. That is result of genotypic ratio. As we just read, 1 RR, 2 smaller, capital R smaller, 1 smaller, capital phenotypic, 1 red, 2 pink, 1 white, right? So here in incomplete dominance, what we get is all the 4 being pink in color. That is crossing of red and white flower gives us pink color, which is termed as incomplete dominance. That is neither red or pink is being dominant. Now, second type is what? Codominance. Now, what is this codominance? In codominance, both the alleles or genes of an allelomorph pair express themselves equally in F1 hybrids. That means both the character or both the genes are expressed together. In the earlier one, incomplete, both are not expressed. Here both are expressed. So such alleles which are able to express themselves equally independently in hybrid are called codominant alleles. So in species or hybrids which are able to express separately or independently, they are called as codominant alleles. Thus in codominance, both alleles are expressed. Classic example of codominance is coat color in cattle. Coat color, there's a skin color, as we can see in this image. When red cattle RR are crossed with white cattle WW, where it's capital, both F1 hybrid capital R capital W are Rowan. So the name given is Rowan for the crossing of red cattle and the white cattle. So the Rowans have mixed of red and white color hair. Thus both traits are expressed equally. In F2 generation, red RR, Rowans RW and white WW are produced in the ratio 1:2:1. Thus in codominance, genotypic and phenotypic ratios are identical. So as you can see here, phenotypic ratio red, white, genotypic capital R, capital W, the gametes row 1 RW, the gametes again capital R, capital W. In the F2 generation, when crossing takes place, one pure red RR, one RW, second RW, that is row 1, 2 in number, and fourth one WW capital, which is 1 white. So the genotypic ratio is 1:2:1. So that is a representation of codominance in cattle. So we can see the difference in incomplete and codominance. Now coming to multiple alleles, the third type, that is more than two alternative forms or alleles of a gene in a population occur the same locus on a chromosome or its homologue are known as multiple alleles. So here what is said, the two alternative forms of a gene in a population which occupying the same locus or the position in a chromosome. Multiple alleles arise by mutation of the wild type of genes. So when you say multiple, more alleles are or factors are seen. And this reason mutation. What is mutation? Spontaneous changes which occurs maybe due to some chemical reaction or some because of UV rays, etc. A gene can mutate several times, producing a series of alternative expressions. So it can keep on mutating many times, as we can see in the present situation. Different alleles in a series show dominant recessive relation or may show codominance or incomplete dominance among themselves. So here it can be codominance, it can be incomplete dominance or dominant recessive relationship, as we seen in the earlier two cases. Wild type is dominant over all the other mutant alleles. Now the term mutant, I think it's very clear in the present scenario, like you can see in case of the COVID variant, where you can see different mutant virus keeping on changing, right? Where one may be dominant, other may be not dominant, but it can cause severe problems, right? So in Drosophila, a large number of multiple alleles are known. Example, the size of the wings from normal wings to vestigial wings, that is no wings, that is just stumps is due to one allele, that is VG, in homozygous condition. The normal wing is wild type while the vestigial wing is recessive type. There is another example. Good example of multiple is the ABO blood grouping in humans, where ABO or AB group is found. So here the table 3.3, few phenotypes and genotypes in Drosophila. As I mentioned, what is Drosophila? You can use a simple name, fruit fly, which is usually observed in the fruits if it is kept for a longer time outside. Then phenotype, normal wings. Genotype, we can see VG plus. Nicked wings, we can see VG, NI. Notched wings, VG, NO. Strap wings, VG, ST. And vestigial wings, VG. Okay. Now moving further, image and another example for you to understand, which is out of the textbook, multiple alleles in case of rabbit coat. So phenotype, coat color, allele, pattern of inheritance, dark grey, chinchilla, Himalayan, white, which is given as capital C, small c, ch, small c, h, small c. Dominant to all other alleles, which is dark grey, dominant to Himalayan and to white, dominant to white, and recessive. So you can see the possibility of genotypes or the phenotype. You can see dark grey with the image I put up, chinchilla, which you can see the coloration, light grey, Himalayan, and albino type. So there can be 5 type of multiple alleles seen. So this example I just added for you to have a better understanding of what is this multiple allele as such, other than the example given in the text. Then you can see a sickle cell anemia, where the usual spherical shape of the RBCs in case of sickle cell becomes C shape, or the sickle shape anemia. Then fourth type is pleiotropy. Now that is what we just saw, sickle cell anemia. When a single gene controls 2 or more different traits, it is called as pleiotrophic gene. What is pleiotrophic? When single gene controls 2 or more different traits, and the phenomenon is called as pleiotropy or pleiotrophism. The phenotypic ratio is 1:2 instead of 3:1 because of death of a recessive homozygote. So here it is not 3:1, it is 1:2. Reason, the recessive trait eventually dies. The disease sickle cell anemia is caused by a gene HBS normal or healthy gene HBS dominant. So normal gene is usually dominant, the sickle cell gene is HBS. The carrier heterozygous HBS show signs of mild anemia as the RBCs become sickle shape, that is half moon shape, only under abnormally low oxygen concentration. The homozygous or homozygotes with recessive gene HBS, however, die of total anemia. Thus, the gene for sickle cell anemia is lethal in homozygous condition and produce sickle cell trait in heterozygous carrier. 2 different expressions are produced by single gene. Now here, as when crossing over takes place, one will be the dominant one, 2 will be the you can say a carrier, and the fourth one will be the recessive, and the recessive, as you mentioned, does not survive. So a marriage between 2 carriers will produce normal carriers and sickle cell anemia with children in 1:2:1 ratio. But the sickle cell anemics die leaving the carriers and normal in the ratio 1:2. Also that the 1:2:1 is not depicted because that one anemic sickle cell condition does not survive. The heterozygotes or carrier can be identified by microscopic examination of the blood. So it can be identified by microscopic examination. Now here are a few images from the textbook and 2 other images which I added for better understanding. First we can see the image from the text. It is figure 3.14, representation of pleiotropy, parents, phenotype, sickle cell carrier, the gametes, genotype, HBA, HBS. As we just read out, HBA normal gene, HBS carrier, HBA again here, and HBS. When the crossing over takes place, the HBA from either parent will be normal. 2 sickle cell carrier and one anemic. So 1:2:1, and the one which is the anemic condition recessive trait does not survive. Here I added two simple images, the single gene pleiotropy, multiple traits. So trait A, trait B, trait C. So as we read out, one gene is responsible for two or multiple traits, which is termed as what? Pleiotropy. So that's why I added this image to make you understand. So beta globin gene mutation, that is for sickle cell anemia, single gene pleiotropy, it can be responsible for blindness, for liver failure, for heart attack. So it can have multiple traits, trait A, B, C, as I shown in case of examples of 3 organ which can be affected by this single gene. Lastly, the assignment question based on today's part of the chapter or the video. Clear? The remaining two parts I'll be adding very soon. So thank you all of you.